Good afternoon, everyone. First, I would like to thank the Belgian Society to give me the opportunity to present you the first results of my thesis, which concerns the study of the hippocampal synaptic plasticity and neuroinflammation during the course of experimental autoimmune encephalomyelitis. Synaptic plasticity is the ability of neurons to modulate the strength of their synaptic transmission after an electrical activity. This process is used in the hippocampus to ensure long-term memory consolidation. It allows to modify the dynamic of neuronal circuits thanks to structural and functional remodelings at the level of synapses. Synapses are not only composed by a pre- and a postsynaptic neuron, but glial cells are now considered as the third element of synapses, contributing to neuronal activity. Microglia are part of the innate immune system of the CNS, and astrocytes actively contribute to immune responses. A complex crosstalk takes place between these glial cells and neurons to ensure neurotransmission and neural plasticity. It is well known that the immune system plays a physiological role in learning, memory, and synaptic plasticity. However, during an inflammatory event, this system can be rapidly disrupted and can alter neuronal network activity, leading to cognitive impairments. These disorders are very common in neuroinflammatory diseases, but the mechanisms implicated in these deficits remain purely understood because of the large diversity and complexity of immune responses that can be engaged. With this view, our project aims to study the effects of neuroinflammation on neuronal network activity and synaptic plasticity of the hippocampus in order to bring out the inflammatory mechanisms implicated in cognitive impairments during neuroinflammatory disorders. For that, we used an experimental model of CNS chronic inflammatory disease, the experimental autoimmune encephalomyelitis. This disease develops as the result of an inflammatory autoimmune reaction against the myelin shifts of neurons, leading to axonal demyelination and to motor disturbances. The induction of EAE requires 2 subcutaneously injection of a myelin antigen, the myelin oligodendrocyte glycoprotein, and 1 intraperitoneal injection of the pertussis toxin. Thanks to a scale allowing to score motor symptoms, we are able to follow the disease progression with time after the immunization. EAE mainly develops with a relapsing-remitting course, which is marked by an ascendant paralysis followed by a partial recovery of motor symptoms. Unlike other studies, we were interested in the different steps of the disease. So, our analyses were realized during the peak of the disease, or the stage 3, and also during the short and the long remission stage. We also made a difference between male and female mice. The activation of glial cell was followed during the disease course with a fluorescent immunostaining of astrocytes and microglia with the biomarkers GFAP and IBA1 marked respectively with a green and a red fluorescence. We also analyzed the hippocampal synaptic plasticity during the course of the disease with ex vivo electrophysiological recordings made on acute hippocampal slices. We studied a particular form of synaptic plasticity, which is long-term potentiation. Our LTP measurements were made at the level of synapses between the Schaeffer's collaterals and the C-RON region of hippocampal slices that were kept alive in a recorded chamber with oxygenated artificial cerebrospinal fluid. Our immunostainings made on male EAE mice reveals that the number of both activated astrocytes and microglia follows the disease progression, as it enhances during the peak of the disease and then it decreases during the remission stage. However, this evolution is more pronounced for microglia than for astrocytes. the enhancement of activated microglia is more important than astrocytes at the peak of the disease. However, the number of activated microglial cells continues to decrease during the long remission stage compared to the short one and remains higher than the control level. This is not the case for astrocytes, as no distinction was found between the two remission phases and with the control situation. For female mice, the enhancement of activated glial cells is similar between astrocytes and microglia at the peak of the disease. However, the reduction observed during the remission stage is not so obvious. The number of activated astrocytes is significantly lowered during the short remission stage, but not during the long one, which seems higher. For microglia, no statistical difference was found between the two remission phases and with the stage 3. So, in contrast to results obtained from males, the activation of glial cells during the course of the disease in female mice is not so clear. We then compared the inflammatory state of the hippocampus between males and females. Concerning the number of activated astrocytes, similar levels were obtained between the two sexes for any stage of EAE. We can also note that the activation of astrocytes is exactly the same between male and female control mice. However, for microglia, the trend is different. Unless some insignificant results, we found a higher level of activated microglia during EAE. Moreover, there is also a higher level of activated microglia for the female controls, so suggesting a higher baseline inflammatory level in the females compared to the males. We then analyzed the hippocampal synaptic plasticity thanks to LTP measurements. In the male mice, we found a higher level of potentiation during the peak of the disease compared to the control situation. More interestingly, we found that the LTP was reduced to the control level during the short remission stage, but it continues to decrease when the remission is longer extended to reach a level significantly lower than the control one. This suggests the time-dependent impairment of the hippocampal plastic potential during the remission course of EAE. For female mice, similar results were obtained, with a higher level of potentiation during the peak of the disease and a lower one during the remission stage compared to the control level. We then analyzed if the reduced synaptic plasticity that we found during the remission stage is not associated with a cognitive impairment. For that, we used a behavioral test, which is the contextual fear conditioning. This test was made on remitting mice either 1 hour or 24 hours after the training session to evaluate respectively the learning abilities or the memory of these mice. The test revealed no alteration of the learning abilities of remitting mice as the freezing level increases significantly. However, when we performed the test 24 hours after the training session, a lower freezing level was obtained, suggesting a memory deficit in remitting mice during EAE. In conclusion, we can say that although EAE is a pathology mainly characterized by motor disturbances, we also found that the hippocampus is affected during the disease. An inflammatory state marked by activated astrocytes and microglia develops during EAE and follows the disease progression. Moreover, modifications of synaptic plasticity were found, but they are inversely correlated to motor symptoms. During the peak of the disease, a complete hind limb paralysis is observed with a higher inflammatory level in the hippocampus, but the hippocampal synaptic plasticity was improved. However, during the remission stage, when a partial recovery of motor symptoms and a lower inflammatory level were obtained, a cognitive impairment was found, along with reduced synaptic plasticity that seems to be time-dependent. Future experiments will consist on a more detailed analysis of the structural and functional modifications induced by inflammation in the hippocampus during EAE, and an analysis of the individual contribution of astrocytes and microglia to hippocampal synaptic plasticity during an inflammatory event. Finally, I would like to thank my colleagues for their support and also the FNRS to finance this project. Thank you for your attention.
Okay, thank you for this nice presentation. Are there questions?
Hello. I was wondering which type of stimulation protocol do you use to induce LTP, because you didn't mention it, I think, or maybe I missed it, and you didn't indicate it, so I was just curious about the type of stimulation protocol. For inducing LTP, yeah.
So we induce a high-frequency stimulation,
high frequency
100 hertz during 1 second.
Just once?
Yes.
So once?
One train.
Okay. Yeah, because I saw the magnitude reaches like, 220%, something like that.
Yeah.
Yeah. Okay. Thank you.
Other questions?
So I was wondering about the possible variability, especially in your LTP experiments, because in EAE you have lesions, sites of the brain that are infected, have big inflammation and demyelination, and other sites not. And I know that sometimes the hippocampus is affected, and in some animals that have the same EAE score, the hippocampus is not affected. So I'm surprised by seeing that the variability on the LTP, which is a local circuit, is very, very low. You would expect in some animals a big effect, and in other animals that don't have a lesion in their hippocampus, a small effect. Do you see variability, or is it just the inflammation that you expect and not the demyelination that has the effect on the LTP?
In fact, we think that there is no demyelination in the hippocampus during EAE, because we had performed some MBP immunostaining before, and if you want, I have the results. So as you see, we can see that the MBP immunostaining is similar between the controls and the different stage of EAE, between the peak and the remission stage. So we think that there is no demyelination, so the LTP variations that we see cannot be due to demyelination. But we need to quantify our MBP immunostaining to confirm these results.
So, and did you also in the IBA1 stainings did not see an increased lesion sites in the hippocampus? Did you not notify this?
No.
Other questions?
It's related to my previous question, because I was wondering then which percentage of the synaptic strength do you use to induce LTP then?
Do what?
For the input-output curve, which percentage of the maximum stimulation strength do you take?
We stimulate between 2 and 13 volts.
Yeah, but okay. No, because I was wondering if you take 30 or 35% of the maximal response during input-output curve.
or
40%. 40%.
Yeah.
Okay. This might be the reason why, because
Yes.
I was just wondering why, when we use, for example, theta-wave stimulation or once high-frequency stimulation, the magnitude is the maximum slope of the LTP is much smaller than the one that you found. And it might be because you use 40%.
Yes.
Okay. Yeah. Just thank you.
Okay, thank you. Then we will go on with the next speaker.